Hi, my name is Bryce Barbee, and I'm a junior studying ecology, behavior, and evolutionary biology in the Pinter-Wollman lab at UCLA. So during my time here, I've been conducting my own research project regarding how nutritional needs and predation risk combine to shape foraging decisions in ants. So a main concept in my project involves trade-offs during foraging, and this is because when considering the ability for an individual to forage safely in order to promote their best chance of survival and highest fitness gain, risk assessment is a major factor. So during foraging periods, it's often easier for individuals to become open to predation and other risks, which leads to a trade-off system that comes for the need for safety and food while foraging. This relates to the idea of optimal foraging theory, which talks about the decisions that individuals must make when foraging that maximizes the net energy gain for the individual. Combined with the ideas from before about maximizing the net energy gain during foraging, the influence of hunger also plays a role in risk judgment as well. It's been shown that hungry individuals generally lead to riskier individuals when it comes to finding food. And in my project, I'm interested in seeing how this idea is upheld in a situation of group level hunger, where individuals forage together at sites for the benefit of the whole group. So with this, I will use Argentine ants because they are good models to test this idea due to their very large colony size and social foraging behaviors. My project also takes into account the differences in nutritional needs of Argentine ants. And so one of the main parts of their diets are sugars in the form of carbohydrates, which they use as the main energy source for the workers to carry out tasks. And proteins are also essential for Argentine ants, as they use these proteins that they forage mainly for brood growth and development. With this, my project looks at what nutritional factors affect foraging under risk in Argentine ants. And the first hypothesis is that nutritional deficiencies will affect a group's decision to forage when risk is present. And so the first prediction for this is that nutrient-deprived groups will be more likely to forage in the presence of risk. And the second hypothesis is that the food type that the group has deficiency in will determine its decision on what to forage when risk is present. And so first, I believe that groups that have been deprived of sugars will be more likely to forage for sugar in the presence of risk. And then groups that have been deprived of proteins will be more likely to forage for proteins in the presence of risk. Here's the housing/foraging area setup that I made to run the experiment. And so I made a simple schematic to show the setup where I'm using 3 different cups for 3 different areas. And so first is the housing cup. This is where the ants are kept before the trials begin. And connected to the housing area by tubes are 2 different foraging sites, one for the protein foraging area and one for the carb area. And then during the period before trial starts, the 2 foraging areas are blocked off using cotton balls, as shown so that the ants can't get into the different areas. Then during the testing phase, the cotton balls are removed, allowing the ants to enter the different foraging areas through the tubes. For the trials procedure itself, I started with a food deprivation period of 2 days, in which the ants were starved according to their treatments. And then after the 2 days, I added the carb-based food and the protein-based food into their respective foraging areas. And the food was placed on a piece of filter paper, and on the paper was also the risk that I used for the experiment, which was 2 drops of formic acid on both sides of the food. And formic acid was used here because it's a defense mechanism used against the Argentine ants by different formicine ant species, and so they would count this as a risk. Then I removed the cotton balls that were blocking the tubes, and this allowed the workers to access both of the foraging areas. After this, I recorded for an hour at 5-minute increments the number of workers on the food, in the food site, and in the tubes to the foraging areas. In order to test my experiment, I had 5 different treatments that I used. And so the first treatment was the no starvation with risk treatment. And this one, the workers were not deprived of any food, and they were allowed access to proteins and carbohydrates during the 2-day period. And then risk was present at both of the foraging sites when they were allowed to go into them. Then I had the deprived for carbs treatment, where they were not allowed carbohydrates, but were allowed proteins, and risk was also present at the sites. Then I deprived them for proteins, and risk was present at the site as well. Then I had a treatment where I deprived them for both with risk present. Then finally, I had a treatment where I deprived them for both with no risk present at the foraging sites. And I included 6 replicates for each of these treatments with 300 workers in both. So for some results for mine, the first one is looking at comparing the counts of forager recruitments for carbohydrates and protein areas for the different treatments. And so this first plot is showing the counts over time, where on the x-axis is time and on the y-axis is forager recruitment counts. And on this, the pink is showing the recruitment to the carbs/sugar area, and the orange is showing the recruitment to the protein area. Each shape in this represents a different replica, and the mean line is shown between each in the shaded regions. For this first one, this is the control with no starve treatment. And as you can see, there's a slightly greater carb increase over time and a plateau at the end. And then this next one is the starve for carbs treatment. And this one has a much steeper increase for carbs, and they didn't care about protein recruitment as much. The third one is the starve for protein treatment. This one starts slightly more recruitment for the carbs, but ends with protein recruitment being higher on average. Then there's one to starve for both, where it looks similar to the starve for carbs treatment, and that carbs were favored and proteins seem to not be cared about as much. And then the starve for both with no risk treatment, where there was not a huge difference between protein and carbohydrate recruitment, and even though the carbs seem to be slightly favored, and then there's a lot of variance in the different replicas with a slight plateau at the end of the data. This next plot is looking at as comparing proportions of ants in the carb site for different treatments in the last time point. And so in this, the dotted line is at 0.5, which means the no preference line, and above that line, it shows where carbs were preferred, and below that line, proteins were preferred. And as you can see in this, the first 4 treatments show favored carbohydrate recruitment, and this is greatest when they were starved for carbs and starved for both with risk present. And then in the last treatment, it's somewhat equal with no great preference towards either carbohydrates or proteins. A and B in this show the treatments that were statistically significantly different from each other. And so the only significant ones here were the starve for both and the starve for both with no risk. And in this, there was a much greater preference for the carbs when risk is present. So for a summary of the results, first, there's a general preference for carbohydrates across the different treatments. Next, there's a greater preference for carbohydrates when starved for carbs and when starved for both with risk involved. In this, there was a less of a preference without the risk involved. And proteins were generally not prioritized throughout the different treatments, with the strongest recruitment for proteins being when there was no risk present at the foraging areas. For future work, I would like to add some ecological context to my study. So the next one that I want to look at is how does group size affect foraging under risk with varying nutritional deficiencies? And the idea with this one is that if there are more foragers, then there will be a higher carbohydrate demand. And then the next question that I want to look at is how does the presence of brood affect foraging under risk with varying nutritional deficiencies? And for this one, I would like to see if more brood will equal a higher protein demand. So my work showed results describing how nutrient deficiencies and foraging behaviors are intertwined. And with this, it was also shown that both nutrient types were not prioritized equally, which affected the ants' willingness to forage in the presence of risk. And using these concepts, I believe it'd be interesting to see how the idea of unequal nutrient priority is potentially demonstrated across different types of animal species, and looking into the differences between species with social and solitary individuals. And I also believe that it'd be interesting to explore these concepts in different ant species, especially ones that have different colony compositions, such as group size. So with this, I would like to say thank you to Dr. Pinter-Wollman for allowing me to conduct my own research in her lab, and also for guiding me through all the different steps involved during my project. And I would also like to thank everyone in the lab for the feedback and all of the help that I've received throughout my project. And thank you all for listening, and I hope you enjoyed it.